drugs. So drugs does not mean purely only on the medical line. This also means many things in the food, then the other cosmetic items and other thing. So in such type of workshop or seminar, you will be given a chance to explore in high performance computing system, where our main speaker, Dr. Saurabh, is going to explain about the high performance computing system that we have and what he is doing, and also many things about the problem solving areas. What are the difficulties that you have? And from our side, what we can say is we have a lot of related publication in this area, and some of our labs are set up to do this activity. And also we have designed many courses and many expertise in the same area. So I want to welcome all the participants here. You listen to the topics or the delivery of our experts of today. And once you have some common interest area, then definitely we can expand this type of workshop to a week or a month or other activity. And there are a lot of problems that we have to solve together for the betterment of our society. So I want to welcome everybody to come with a problem, and then let's try to find out a solution jointly. So once again, I want to welcome, and I want to grace the this function, and I want to declare that this function is digitally open, and we are ready for the next events, and want to wish the organizing team all the best. Thank you.
Thank you, sir. Now we'll have the digital lamp lighting. So it is our immense pleasure, senior professor, senior biotechnologist of this entire country, Professor Probodh Borah sir, is with us here today. So he will also deliver the keynote address. But before that, I would like to request Professor Borah sir to address the gathering. So he is basically the heading the Department of Animal Biotechnology in the Veterinary College under Assam Agricultural University currently. So over to you, sir.
Thank you, Dr. Mohanto. Respected Director, NIELIT, respected faculty members of NIELIT, Dr. Panka Setia, invitees, and the participants of this webinar. So I'm very happy and feel privileged to get associated with this important webinar organized by NIELIT. As you know, in the modern biology, bioinformatics has become a very important part, and it has been introduced extensively in both undergraduate and postgraduate curricula of almost all the universities of the country. Although this particular discipline was not known when we were students in the undergraduate or postgraduate level in till 1980s, but the recent developments in modern biology, biotechnology has necessitated the learning of bioinformatics tools and techniques for every student who aspire to do some research in any field of biology. Now, bioinformatics is an integral component of modern biology, both in basic biology as well as in applied biology. But To be specific, in this particular discipline, even today, there is dearth of expertise in the country compared to some other developed countries, and also in comparison to our neighboring China. In fact, in most of the advanced countries of the world, in the modern biology and biotechnology laboratories, if you visit, you will find Chinese bioinformaticians are leading in most of the labs. Although the journey in bioinformatics in India started way back in 1986-87 with the initiative of the Department of Biotechnology, Government of India under Ministry of Science and Technology. But still, we are lagging much behind in this particular area. And I think much effort is needed, both from the level of the government as well as from the academic institutions, particularly universities, to build necessary manpower and expertise in this particular field so that globally we can compete with the rest of the world. And also, we can also host the important biological resources in the field, in the form of biological databases of global, so to say, strength, so that we can lead in this field in future. So in this aspect, I think the effort taken by NIELIT Guwahati in training the students and other aspiring participants in the field of bioinformatics by way of conducting not only this kind of webinar and training, but also conducting diploma certificate examinations and strengthening the bioinformatics infrastructure in their own institute, and also the regional centers located in different parts of the northeastern region in the last few years. Particularly, I'm very thankful to Dr. Saurabh Mohanto, whom I know that he's one of the pioneers in this field in the northeastern region, because at the time when he started learning and teaching bioinformatics, I think it was not known to many of the senior teachers, faculties in the universities. And in fact, I'm happy to see that many of his students in the first few batches now have already been appointed as teachers and faculties in different institutions and universities, not only in this region, but also outside the region also. We are in close touch in the last more than 10 years in doing activities of this kind, organizing seminars, webinars, as well as organizing short and long-term training programs for the benefit of the faculty members and students in different areas of bioinformatics. So today, they have chosen a very important topic in the area of bioinformatics, that is computer-aided drug discovery. So this will be only a sensitization program of 1 day, but nobody can learn bioinformatics and become a master in attending a webinar of short duration. That we must have to realize and agree. But this is an eye-opener for the students and participants. And I think this will sensitize them to go into depth of this particular field in bioinformatics in future. And this will also motivate them to venture into learning bioinformatics in future, so that they can also use the learning from this kind of webinar and training in their own field of biological research. So that will be very helpful and prove to be fruitful for augmenting their research efforts in their own field of research. So I hope that this will be very fruitful for the participants, and they will carry forward the message that the team wants to put forward through this webinar so that more awareness and interest is generated among the students community and the teaching fraternity of Northeastern region and also outside the region in the area of bioinformatics, so that India can emerge as one of the leaders in this particular emerging field. So with this brief note, I'd like to conclude. Thank you very much for giving me the opportunity to share some of my views in this inaugural speech. Thank you.
Thank you, sir. Thank you. So we are very much privileged to have with us today, sir. So again, we will hear from you in the keynote address with after a very short time from now. So now I would like to introduce and welcome Professor Robenta T. He is the professor in the Department of Botany by Guwahati University, and as well as the Director of Internal Quality Assurance Cell of the university. So I'd like to say a few words, sir, to the participants.
Okay, thank you. Very good morning to everyone present here. Director NIELIT, Guwahati and Shillong, Professor Yumnam Jayanta Singh, Dr. Saurabh Mohanto, today's keynote speaker, respected Professor Probodh Borah, distinguished resource person, participants, ladies and gentlemen. Thank you for the opportunity, and I am privileged to participate in the this inaugural program of the national level webinar on topic is Principles of Computer-Aided Drug Design. Already Professor Borah sir has given an idea regarding the today's topic. So the friends, we meet a time now when information and communication technologies has become very much essential for societal development as well as economic development around the world. The whole world is talking about now the discovery of new drugs, the vaccines, and now more than ever, the biotech and pharma industry is in the spotlight. The drug discovery process involves millions of dollars, elaborate and time-consuming research, and researchers have been experimenting to make this process more efficient, to cut down the time involved and cost involved, basically. The recent advancement in bioinformatics, as Professor Borah told, and also together with the cheminformatics have led to some major breakthrough in drug designing and discovery processes, and one such technology is computer-aided drug design, the today's topic of this national level webinar. And under this, some of the important domains like how to make effective implementation of analytics in various stages of drug discovery, including drug repositioning, then target identification, predicting drug combinations, and so on. So we have the resources, and besides the plant resources, we should also be more focused to explore whatever we have, the traditional resources also, which are expected in the form of mouth to mouth from generation to generation. And that is more important to make the database and provide the implementation of this information technology so that these resources, we can preserve it for our future, the days to come. And to make the translational research to how to translate these traditional resources into the fruitful research. And I sincerely believe that whatever the team under the leadership of the efficient director, Professor Singh, is doing, this really commendable. And so far, my knowledge, I know from the very beginning of this DOEC society and afterwards the NIELIT, they are doing really wonderful for the people of the region. They are reaching all the target groups, starting from through the faculty development program, even through the training program, to the office people, to the industry, to the and so many. And as Professor Borah already told, that the one-day webinar will be just a glimpse how to people, the participant will be able to understand about the concept, and that should be carried forward in the future research only. And I again believe this today one-day webinar will be very effective one. A very good number of participants I can see here that have joined from not only from the country, even from the abroad. And I wish all the best for a successful program. And with this, thank you, Dr. Saurabh Mohanto, for giving me this opportunity, and Dr. NIELIT. Thank you so much.
So thank you so much, sir. So yeah, we are very much privileged to have with us today, sir. So we will again, if opportunity comes, we will again hear you. So now I would like to request Dr. Panka Setia. So he is one of the he is basically one of the pioneer in bioinformatics along with our team. So he is basically currently serving as a faculty member, as an assistant professor, senior assistant professor in the Department of Life Sciences in Guwahati, Dibrugarh University, which is another premier university of this region. So I'd like to request Pankaj to the participants.
Thank you, sir. A very good morning, respected Director NIELIT, Professor Probodh Borah sir, Professor Bhaven Tati sir, and other dignitaries and participants present in this webinar today. So, as I know, NIELIT Guwahati is the pioneer of teaching institute in the entire northeastern region, and I am one of the alumnus of that institute. At that time, the institute was known as DOEC Center Guwahati. It was DOESCC, DOEC Center Guwahati. So when NIELIT started this course, Bioinformatics A level, way back in 2003 and 2004, I think most of the universities in the Northeast were not aware of the subject very well. So in the course curriculum of different MSc subjects, the bioinformatics was not a part of the subject. So NIELIT has taken NIELIT has always played a key role in creating awareness and generating manpower in the field of bioinformatics in the northeastern region and as the country as a whole. So I am very happy that NIELIT is organizing this one-day webinar on this computer-aided drug designing tools and techniques. And I wish the webinar a grand success, and I'll be happy to interact with you in the technical session, in the afternoon session. Thank you.
Thank you, Dr. Setia. So we'll again have an opportunity to hear you in the and now I'd like to request our senior officer, scientist from National Institute of Electronics and Information Technology, Guwahati, Rintu Das sir, to say a few words about NIELIT and also to give the vote of thanks to the to end the inaugural session. Rintu sir, please.
Good morning, everyone. Respected Professor Probodh Borah sir, respected Professor Bhaven Tati sir, respected Director NIELIT Guwahati, respected Dr. P. Setia, all the participants here, and our my colleagues of NIELIT, National Institute of Electronics, NIT, Guwahati. NIELIT, National Institute of Electronics and IT, is the HRD arm of the Ministry of Electronics and IT, Government of India. The mandate of NIELIT has been to provide various courses in the field of ICT, mostly in the non-formal sector. And gradually, we are also moving into the formal sectors. Like we are offering formal courses and graduate level, PG level, MSc. We are offering MSc course. Then we are offering BCA level courses. Then some of our NIELIT centers are offering MCA level courses. Then a few centers are offering MTech level courses also. We have formal courses as well as non-formal courses. Then NIELIT Guwahati, as already mentioned by the eminent speakers, NIELIT Guwahati has been involved in this offering the bioinformatics course for the last about 12 years, since 2002. And it has been mostly taken up by Dr. Saurabh Mahanta. The courses, as mentioned, we have our alumni are working in the various reputed MNCs in the pharmaceutical sectors and computational biologies. Many are doing research works in the field of bioinformatics. Presently, under the guideship and leadership of Professor Dr. Yomnam Jayanta Singh, NIELIT Guwahati has been handling various level courses, especially in the emerging areas like cloud computing. Then we are moving ahead with the blockchain technology, then machine learning, artificial intelligence. And I'm really happy to be a part of this today's national level workshop on principles of computer-aided drug design. And I'm really honored to quote the vote of thanks in this inaugural session. I sincerely thank Professor Borah sir. He has been associated with NIELIT for so many years I have seen. Thank you, sir, so much for being here to give the motivational talk and address the participants. Thank you, sir. I sincerely thank Professor Bhavin Tati, sir, as the professor, Department of Botany, and Director, IQAC, Guwahati University. Thank you, sir, so much for highlighting the various drug designing process. And I sincerely thank the director, NIELIT Guwahati, Professor Dr. Y. Jayanta Singh, sir. Then I thank Dr. Setia, P. Setia, sir, for being here among us. And he will also be taking a technical session in the afternoon. And at last, I would like to thank all the participants for patiently being here among with us and taking part in this national level event. Thank you once again. Thank you very much, all of you. Thank you. Over to
so thank you, sir. Thank you. Now it is time to listen to the vast experience of Professor Probodh Borah sir in the form of a keynote address. So I request Borah sir to present the address. I'd like to give a brief introduction about sir, although it is not very easy to introduce a stalwart in physical sciences in just few words, but I'll try my best. So, sir, Probodh Borah sir, is a professor and head Department of Animal Biotechnology, College of Veterinary Science, Assam Agricultural University, located in Khanapara, Guwahati. So, he did his PhD in 1994, the postdoc from United States of America in 2010, and he has till now he has 35 plus years of teaching and research experience, and he has been one of the very active scientists in the field of bioinformatics. he helped the setting up of the bioinformatics infrastructure facilities across the northeastern region, funded by Department of Biotechnology, Government of India. Then he is basically the member of the committees of other universities and institutions like IIT, different IITs, Borah University, Cotton University, ISST, NEHU, Guwahati University, so GMCH, Mizoram University, Don Bosco University, many colleges and other universities of India. So he also headed the state level biotech as the state coordinator, and he is basically a scientist, a researcher, a professor, and the NIELIT is very much fortunate to have him with us today. So with this, I'd like to request Borah sir to present the keynote address, and all the participants are requested kindly to listen to sir. Thank you. Over to you, sir.
Thank you, Dr. Mohanto, for introducing me to the participants and saying about me with some sweet words. Thank you very much. So now I think my presentation is visible to all of you.
Yes, sir. It is visible.
Okay. So the topic of this particular webinar is computer-aided drug discovery. So I shall give a brief introduction to the topic, an overview, but detail about the topic will be discussed in 3 different lectures to be delivered subsequently by eminent experts in this field. I shall be only giving you a brief introduction to the topic and what are the important steps and processes involved. But regarding the procedures and tools and techniques, I shall not describe in detail. The 1965 Nobel laureate in physics, Richard Feynman, once stated, if we were to name the most powerful assumption of all, which leads one on and on in an attempt to understand life, it is that all things are made of atoms, and that everything that living things do can be understood in terms of the zigglings and winglings of atoms. Although at that point of time when he spoke about understanding the biological processes by understanding the zigglings and winglings of atoms like that of material world was not acceptable to the biologist. But later on, with the advent of omics technology in biology, after the initiation of the field of genomics, with the advent of the genome sequencing, it is now realized and accepted by one and all that like the material world, the principles and the procedures involved in material sciences can be applied widely for understanding the biological phenomena. Really speaking, this statement of the Nobel laureate is applied today in the field of bioinformatics to a great extent to explain most of the important biological phenomena based on the interactions between biomolecules. As the students might be aware that we have got 4 important biomolecules which play a very vital role through their intimate interactions to give all the functionalities of the biological system. Proteins being the primary unit of life, both structurally and functionally, it is the unit of life. It is also assisted by other molecules like nucleic acids, DNA and RNA, carbohydrates, and lipids. But of course, there is also a role to be played by macro and micro elements, minerals, vitamins, hormones, and many other molecules. And with an intimate interaction among the biomolecules, it generates certain pathway of interactions that lead to the biological activity, or that leads to the entire biological system. So bioinformatics is the discipline in which the knowledge of material sciences like physics, chemistry, mathematics, statistics, engineering, computer science, information technology are all utilized so as to help the biologist to understand the complexities in biological system. So drug discovery before the advent of bioinformatics was based merely on wet lab experiments. And if you go to the process of drug discovery before the advent of the omics era, for discovery of drug, various steps were involved, and each of the steps were very laborious, time-consuming, and very expensive. So if you look into the cost and time involved in drug discovery before the advent of the omics era, from target discovery to lead recognition and optimization, it took around 2.5 years. Then to reach preclinical trial status, it needed 3 years. Then to go for clinical trials, phase 1, 2, 3, and 3, it took around 1 year. Then thereafter, to get the approval of the agencies, regulatory bodies, it took around 6 years. And finally, to commercially produce the particular compound for use as a therapeutic agent, it took another 1.5 years. So total time required was around 14 years. And interestingly, if around 1,000 compounds are identified as leads at the initial stage of the discovery process, finally, when it goes through the different processes of discovery, only one compound came to the market by the spending of several million rupees to the tune of around $880 million. So now, after the advent of bioinformatics as a discipline, there are many aspects in which, in the drug discovery process, bioinformatics tools and techniques can be widely applied, and that reduces not only the time required for drug discovery, but also it reduces greatly the cost of production of drug. As a result, in the recent time, even within a period of 5 to 7 years, new drugs could be discovered, which are applicable within that stipulated short period of time to get commercialized and utilized for therapeutic purpose. So drug discovery process involves basically 3 important steps: the target discovery, drug discovery, and drug development. First of all, we need to identify the target for development of the drug. That means a molecule in the system of the host or of the microorganism involved in disease process, and to validate the target as the key substance which can bring about changes in the physiological system to restore normalcy. Then after that, we need to identify biomolecules which can interact with the target effectively so that it can either neutralize its bad effect or can augment its helpful effect or beneficial effect. And after that, after the lead molecule is identified, that should be optimized by chemical processes. And then after its testing in vitro at different levels, it is released for first preclinical trial in vivo experiments involving laboratory animals, cell culture, and then finally it goes for clinical trial in 3 different phases: phase 1, phase 2, and phase 3. And ultimately, after successful completion of the entire process, it goes for approval of the regulatory bodies, and after the approval is achieved, it goes to the market. So in the entire process of drug development, there are various aspects involved, and a number of biological and biotechnological applications are required to be applied in every step of drug discovery process. As I said, before the advent of the omics technology, most of these tools and techniques were confined to the conventional wet laboratory experimental methods. But now, many of them are either replaced or supplemented by a number of computational methods. That means many of those tasks can be done now with the help of computer, so that it shortens the time requirement for completing a particular step in the drug discovery process. And also it becomes more accurate and more precise, so that there is no wastage of time and resources for unnecessarily testing a number of probable compounds to discover one lead molecule or to finally come up with a one usable drug molecule. So the computational drug discovery process basically involves 2 different methods. We shall discuss a little bit about that in the following slides. But to describe in a nutshell, we can either approach by identifying the target, and based on the target molecule, we can identify the lead molecule or the biomolecule that can neutralize or can interact effectively with the target molecule because of their conformational similarity. And that can be tested by use of a computational method called molecular docking. So it is nothing but a study of the binding efficacy of the probable lead molecule with the drug target, and to see whether the binding is appropriate to give us the needed effect on the target, either to neutralize it in case the target has to be restored to normalcy by this interaction, or maybe it inhibits the activity of the target molecule in case it is a deleterious molecule of an infectious microorganism. Now, if the docking results are positive, means we are getting a very good interaction between the probable molecule with the drug target, then we can go for synthesis of the molecule, and thereafter in vitro screening. In vitro process, if we get a good result, then we can pass it to the laboratory for in vivo screening, first in laboratory animal models, and then it goes for clinical trials. And finally, as I already said, after the successful results in 3 phases of clinical trial, if it gets the approval of the regulatory authorities, it goes to the market as a drug. But of course, you cannot expect that your results will be always positive in all these steps involved. Most of the time, some optimizations may be necessary before taking the probable biomolecule for an in vitro and thereafter in vivo screening. Now, computational methods are available for screening the biomolecule or the probable lead molecule by a number of different processes. Thereby, you can not only assess the interactivity of the molecule with the target, but also you can see the events occurring after the interaction, the biological activity, and also you can assess the deleterious side effects of the drug by using in silico process. We shall come to that in some later slide briefly, but I think that means in the subsequent lectures, these processes will be discussed in more detail. Then, of course, another approach is to start with small molecules or ligands. There are very important biological databases which provide us now the information pertaining to the structure of millions and millions of different small biomolecules, and they are called ligand databases. From there, you can obtain the necessary information about the ligands or small molecules. And based on these small molecules, with the help of the computational method, now based on our knowledge on the biological activity of some small molecules, we can compare the structure and physicochemical properties of small molecules and can predict the probable biological activity of the molecules and can further test it by the same process of molecular docking with different target molecules to find out the most probable druggable molecule so that it can be carried forward by the same process to the in vitro screening and thereafter in vivo screening. So that means one approach is based on the drug target and the other approach is based on the ligand or the small molecule. I'll not go into detail about each and every step here. Then, as I said, the first and foremost step in the process of drug discovery is the target identification and its validation. Now, a disease may be caused by either infectious microorganisms or due to various non-infectious processes like metabolic disorders or the effect of certain chemical or physical injury or many other non-infectious causes. In case of infectious microorganisms responsible for causing a disease process, the target for drug discovery may be the essential enzymes which are required for survival and growth of the microorganisms. So they are selected in such a basis that they are unique to the microorganism and has got host specificity. Means these enzymes are active only in the microorganism but not in the host, so that if we can neutralize the enzyme by use of a drug, then there will be no survival of the microorganism causing disease, but at the same time it will have got no impact on the host. Similarly, in case of non-infectious diseases, the target can be a native enzyme or a receptor on the cell which are involved in the disease process. So for that, we must have to establish the pathogenesis of the disease in detail, we must have to understand how the disease process advances in the normal physiological system and how the pathological symptoms and the abnormalities in the tissues and organs are created by the disease. So if we understand the entire pathogenesis of the disease, then we can understand about the biological processes involved or the metabolic pathways involved in the disease process. And thereby we can identify an essential step in the required metabolic process which gets altered in the disease condition. And that can be the target of drug discovery. Let me give you an example so that you can understand the process. I'm giving an example of the current pandemic of COVID-19. As you know, it is caused by a virus which has been recognized as SARS-CoV-2 virus, SARS-CoV virus 2. Now, in addition to a number of structural proteins which constitute the structural component of the virus, there are many non-structural proteins. Altogether, 16 non-structural proteins are there, and they play a very vital role both in virulence as well as immunogenicity. For virus survival in the tissues and therefore growth and multiplication, for their replication in the tissues, many of these non-specific proteins are essential for the virus. Although the function of each and every non-structural proteins are yet to be known clearly, but already a number of non-structural proteins in SARS-CoV-2 have been identified, which play a very vital role in the virus replication process. That means without that, the virus cannot replicate. So basically, the SARS-CoV-2 virus produces 2 polypeptides, which are the precursor for production of all the 16 non-structural proteins. So 2 open-ending frames means 2 genes, ORF1A and ORF1B, code for these 2 polypeptides. And these 2 polypeptides are finally broken by, or they are cleaved by, certain proteases, and 2 major proteases, one is called 3CL-Pro and the other is called PL-Pro, which are coded by the ORF1 and ORF1A and ORF1B. These 2 genes, these 2 virus-specific proteases are responsible for cleavage of the polyproteins, polypeptides. And unless these polypeptides are broken or cleaved, the 16 non-structural proteins cannot be formed, which are essential for replication and survival of the virus in the tissues. Now, therefore, we can expect that if we can find some inhibitors which can efficiently inhibit these 2 proteases, 3CL-Pro and PL-Pro, then probably the replication of the virus will be inhibited because there will be no production of the non-structural proteins. So 3CL-Pro is the main protease which is now targeted as the target for the drugs to be synthesized which may be active against the SARS-CoV-2 virus. So this main protease, 3CL-Pro of SARS-CoV-2 virus, is one of the major drug targets for control and treatment of SARS-CoV-2. Now, a drug target, as I said, may be an existing cellular or molecular structure. If we inhibit that, then it can restore normalcy, or it can be an enzyme or a receptor. In fact, most of the drug targets in major diseases are either enzymes, around 50%, or maybe some specific receptors on the cell membrane. They are called Z protein coupled receptors, 23%. So these Z protein coupled receptors, or GPCRs, are the largest and most diverse group of membrane receptors in eukaryotic cells. And therefore, they are common targets for the drugs. But of course, sometimes it may be DNA or RNA also. Now, as I already said, that in computer-aided drug designing, there are two approaches in helping designing and discovering a drug by use of computational process. They are target-based or ligand-based. Two processes are involved: target-based drug designing or ligand-based drug designing. First one is target-based drug design. I'm not going to detail about that. In subsequent lectures, the experts will deal with that. But let me give you a brief idea about that. In target-based drug design, the first and foremost necessity is to know the detailed structure of the target molecule. If we have identified the drug target and validated it as the molecule responsible for causing the disease, then we shall have to identify the target. And there's a message from someone that I'm not audible. Am I audible to others?
Yes, sir. Your voice is clear.
Yes, sir. Yes, sir.
Okay, I think there may be some local problem with some participant. Anyway, so the detailed three-dimensional structure of the target molecule, which are mostly proteins, as I said, either receptor proteins or enzymes. So protein structure has to be known, because as I said, that the initial interaction between the drug and the target molecule is due to the conformational similarity in the physical structure. So therefore, we must have to thoroughly know the detailed three-dimensional structure of the drug target. For that, two experimental methods are commonly used to have the protein structure determination: X-ray crystallography and NMR spectroscopy. But as we know, both these processes have got some major disadvantages and limitations. First of all, they are very expensive methods. The initial installation of the equipment is very high, and also the running cost is also high. But at the same time, the major limitation is that for X-ray crystallography, the proteins are to be crystallized first, but all proteins cannot be crystallized, particularly membrane proteins, which play a very vital role as the drug target, cannot be crystallized easily. And as a result, so far, by experimental process, only around 80,000 cytosolic proteins have been studied by experimental methods to determine the three-dimensional structure, against billions of cytosolic proteins which are to be really experimentally determined. As we know that in a human being itself, there are more than 1 lakh different kinds of cytosolic proteins. But if our capability of determining the 3D structure of protein is limited to the use of the experimental methods of crystallography and NMR spectroscopy, then probably the pace of development in understanding the target structure will be very, very slow. In terms of membrane protein, the situation is much worse, and only around 280 membrane proteins are now so far known. So therefore, to bridge this gap between the necessity and knowledge, we have been left with the other alternative, to use some method to predict the probable three-dimensional structure. And in that, the bioinformatics tools can play a very vital role. Now, computational methods are available, which can give you an almost accurate structure of the proteins, which may resemble to the native protein to the extent of 99% or more. So therefore, even if the experimentally determined molecular structure is not available, the target proteins, if it is known, then it can its three-dimensional structure can be predicted by use of bioinformatics tools. And that process is called molecular modeling or protein structure prediction. There are three methods available for that: ab initio modeling, whole recognition, and homology modeling. In subsequent classes, that will be dealt with little greater detail. Then after knowing the structure of the protein or the target molecule, next the step will be to study the interaction between the target with millions of different probable biomolecules which can have interaction with them. There are now tools and platforms available online in which you can study the interaction between biomolecules, either it is protein-protein interaction or protein-ligand interactions, in a very effective manner. And by using those tools, you can study the binding efficacy between the biomolecules, and thereby out of millions of different molecules available in the databases, you can find out the most probable biomolecules which can have the efficacy of binding to the target molecule in a better way. So there are different steps involved in the process that will be dealt with by my subsequent speakers. And then after the interaction is studied by molecular docking, we can also study the dynamic interaction between the biomolecules by further studying with the help of a simulation model in computer by use of physical principles to elucidate the dynamic interactions between the molecules, their behaviors, their functions, their stability, and their binding affinities by using the molecular dynamic simulations, or MDS. In docking interaction study, we basically look for the conformational similarity between the biomolecules. That means we take into account the physical conformation of the target and the ligand, and we compare the physical conformation to see that there is a similarity in shape and structure. That means we can compare it with a lock and key relationship. A particular lock can be opened by a particular key because of their similarity in conformation in the ridges of the key and the internal ridges of the lock. So this is purely a physical conformational similarity, and that because of that there is an interaction between the two. So in molecular docking, basically we do the same thing. We see the physical interaction because of conformational similarity, and we find out the best fit, the best orientation of the ligand to the target. But of course, the biological molecules do not behave like the material molecules or physical molecules, and their interaction does not limit themselves to the conformational interaction alone. After the biomolecules interact between them, they undergo further changes subsequently over time. It can be compared with a kind of analogy, that is your hand in glove. After you wear a glove, you adjust the glove by moving your fingers, because the gloves are made as flexible and it can be adjusted or adapted to your hands by moving your fingers. So that means after having an interaction between two molecules, the further interactions may be studied over time. If there are changes in the conformation, that may lead to the changes in the biological activity. So that can be done by the simulation studies or molecular dynamic simulations. I'll not go into detail about the softwares and tools involved in that. That will be dealt with in subsequent lectures. So this is a picture showing the interaction, physical interaction between the ligand, a small molecule, and the target molecule. If there is conformational similarity, then there can be interaction between the two. So we search for the best fit by the use of the molecular docking study. Then in molecular dynamic simulation study, we see the interaction that occurs and progresses over time after the physical interaction takes place at the initial stage. So here in this diagram, it is shown that small molecules are binding to a particular target, and after that, the target is undergoing certain conformational changes subsequently after 500 picosecond, and after 1 nanosecond, again, there are more conformational changes. And because these dynamic processes occur over time, that also increases or decreases the interaction between the molecules or affinity between the molecules. So therefore, by doing molecular dynamic simulation, you can study the interactions in a dynamic process so that it can simulate the actual interaction which occurs in the native tissues. So the second method is the ligand-based drug designing. So as I initially introduced to you, it is based on the structure of the ligand or the small molecule. So here the center point is the ligand or the small molecule. That means the key, not the lock. The key is known, and based on the key, we have to identify the probable biological activity of the particular molecule, and then we can use it as a drug for certain specific diseases. So the probable bioactivity of the ligand or small molecule is predicted based on the physicochemical or electronic properties, based on stereoelectronic properties. So the structure is directly related to the biological activity of a molecule. And therefore, we use an approach in this direction to basically predict the biological activity of a small molecule based on their two-dimensional or three-dimensional structure. So this is called as quantitative structure-activity relationship. In quantitative structure-activity relationship, we use the computational techniques to detect the functional group in the compound in order to refine the drug. We refine the drug by both computational process and thereafter by chemical process, so that the drug activity can be increased and the side effect can be reduced. So these chemical modifications, which are necessary for increasing the drug activity or reducing the side effect, can be either predicted by use of the computational process at the beginning and thereafter validating it by the chemical synthesis and modification of the drug molecule by doing the suggested modification. A typical QSAR equation is based on the physicochemical or stereoelectronic properties of the molecules, and this is almost like a regression equation. Activity is predicted based on the particular physicochemical or stereoelectronic properties of the biomolecules. So based on the knowledge of the physicochemical properties and the corresponding biological activity of known compounds, we can predict the bioactivity of the unknown compounds. So these are some of the important stereoelectronic properties that are needed for predicting the biological activity of drug by QSAR approach. They may be related to steric properties, electronic properties, lipophilic properties, or hydrogen bonding properties. Now, after a lead compound is identified, it should be also tested for its drug likeliness or druggability. This was first defined by a scientist, Christopher A. Lipinski, in 1997, based on his observation that most of the orally administered drugs are relatively small and moderately lipophilic molecules. So based on these observations, Lipinski developed a rule of 5. That means he has developed a particular method to identify the druggability of a compound based on certain specific properties, which normally relates to its biological activity without having much deleterious property. So these are some of the Lipinski's rules, Lipinski's rule of 5. I'm not going to detail about that. That will be dealt with in detail by my subsequent speakers. So there are some variants to Lipinski's rule of 5, which have been already advocated by other scientists. And this is one method by which the druggability of a compound can be assessed on the basis of certain specific basic physicochemical properties. Then comes the study of the additional biological properties of the probable drug molecule. After a druggable compound is identified, then it should be further tested for its properties like absorption, distribution, metabolism, excretion, and toxicity. Because the drug must be absorbed in the system, it should be distributed in the body, it should be bioavailable, it should not be degraded by metabolic process very fast, but of course it should be ensured that after some time it is degraded by the metabolic process and finally it is excreted from the system so that it does not remain for a lifetime in the tissues of the host. And also we should also test for the probable toxicity or side effect of the drug. All these are normally tested by the wet lab experiments. In case of conventional process, this takes many months to years to finally come up with a final, so to say, conclusion regarding absorption, distribution, metabolism, excretion, and toxicity of a single compound. It may require more than 3 years, 5 years. But now computational methods are also available by which the software tools can be used to predict all these properties of a probable drug molecule. So computational methods are available by which the ADMET property or ADMETOX properties, ADMET properties or ADMETOX properties can be studied and predicted. So these predictions are also almost accurate and it is very rapid. Unlike the conventional wet lab experiments, which takes around 3 years' time to have the testing in the laboratory, it can be done in only few hours or few days. So this is so fast and robust and so precise, then it gives a very important information to the biologist before taking the drug molecule to the real preclinical trial. Now, after it is done by in vitro processing, when the drug molecule is tested by preclinical trial, as I already said, it will go for further in vivo trial in the laboratory in small animal models, and then it will finally go for clinical trials, phase 1, 2, and 3 in the target host. So in those processes, of course, there is no involvement of bioinformatics or the computational process. So computational process stops once the druggable molecule is identified and it is tested for its ADMET properties. And thereafter, it is given to the biologist for further wet lab testing, in vitro, in vivo, and clinical trial. Now, many a time people ask that whether the computational drug discovery process has been successful. Many people think that this is a theoretical process. This cannot lead to discovery of a drug. The first drug that came to market successfully, which was first designed by in silico process, was dorzolamide. It is a carbonic anhydrase inhibitor, which was first introduced to the market in 1995. And another important product of rational drug design is imatinib, which is a tyrosine kinase inhibitor, which is used against chronic myelogenous leukemia, blood cancer, and acute lymphocytic leukemia. Similarly, there are many different drugs which have been developed against AIDS, like ezinarase and viracept, then influenza-like drug relenza, and many other drugs are still in the clinical trial, or some of them have recently released to the market. So this is practically done within a period of around 5 to 6 years from the computational discovery of the drug till the commercialization of the drug. It took merely around 5 to 6 years when the computational methods are used in drug discovery. So this has reduced the time required to a great extent, and the efficacy of such drugs are also almost guaranteed. Now, major advantages of computer-aided drug discovery is, as has been already explained, the cost savings and the time required to take a drug to the market. But above all, these also help the researchers to acquire deep insight about the drug receptor interactions, and also to understand the detailed pathogenesis process at the molecular level of a disease. So future of computer-aided drug designing or drug discovery is very bright, and it is already giving us very important, so to say, discoveries in the form of drugs against incurable diseases like cancer, HIV, AIDS, etc. But it is an important challenge to the biologist, because although the computational drug discovery process is highly precise and effective, but there is lack of sufficient skilled, experienced manpower to deal with this particular process efficiently. As I already said during my inauguration speech, in India, experts in these lines are very, very counted, and therefore more and more rigorous efforts are necessary at all levels to create adequate manpower so that we can deal with this particular aspect more efficiently, and we can help the pharmaceutical industry to develop more and more new drugs which can be helpful for treatment of emerging and re-emerging diseases in future. With this, I thank once again the organizers for giving me an opportunity to share some of my thoughts on the topic today to the participants. And I also thank the participants for their overwhelming response to this webinar. And at last but not the least, certainly, I especially thank Dr. Saurabh Mohanto for organizing this webinar and also inviting me to deliver the keynote speech. Thank you very much.